Hello and welcome to Health and You. I am Sumi Sukanya Datta. You all would have read about a baby in the US who was suffering from a rare genetic disorder who has got world's first personalized gene editing therapy. This was in order to treat the condition. This was achieved using CRISPR technology or what we also call genetic scissors. It's a matter of great pride and as well as hope that a lot of work is also being done in India related to this cutting edge technology. I'm also very glad to be joined by two very senior scientists who are running two of India's leading laboratories that are working on CRISPR. We have with us Dr. Debojyoti Chakraborty and Dr. Arko Shubhra Ghosh. Thank you so much, Dr. Chakraborty and Dr. Ghosh, for joining us in the conversation. Dr. Chakraborty, we'll first want to understand from you. DNA, as we know, is a code of life. First of all, can you please help us understand how exactly we have been able to join this DNA through genetic engineering? Are genetic engineering and gene editing the same thing, or what exactly is the difference?
Yeah, thank you for having me here. So that's a question that requires a big answer, but I will make it in a very concise fashion. So genetic engineering is essentially making changes in the DNA. Gene editing is a subset of genetic engineering, sometimes also used interchangeably, where we are, essentially trying to make very, very precise alterations in the alphabet that constitute the DNA. Unlike the human, unlike the English alphabet, which has got 26 letters, here there are four letters, but then
Mhm.
they come in different flavors and combinations, which all give rise to some genetic code, which then defines how we are as human beings, right from the color of the eye to the complex, diseases that shape our existence. So scientists have been, it has been possible for scientists to now use certain types of technologies based on molecular scissors like CRISPR, where you could very specifically do some alterations in the genetic code, and then hopefully, in this case of what we are discussing, correct a disorder as well.
Okay. Dr. Ghosh, we would like you to please chip in the conversation now and tell us a little about this particular case and why exactly is it being celebrated as a medical breakthrough all around the world.
Right. So, uh, as Debo stated, so the promise and the purpose for gene editing essentially is that it can make very precise small changes in the genome that can fix a disease. So, the flow, the code of life, like you pointed out, is the DNA is your blueprint and the proteins are your workhorses, right? So, from a small change in the DNA that can lead to profound changes in the protein, which is what causes the disease. So now if you can fix that small change very precisely without changing anything else in the genome,
Mhm.
then you can fix that protein and therefore fix the disease. Now
Okay.
that's precisely what has been done in this case, where they used a base editor. And again, Debo is the authority on that. He can tell you a little bit more about precisely the base editor. And what they did was they took that base editor and they encapsulated it in a delivery vehicle, which was a nanoparticle in this case. And this delivery vehicle then took this editor to the liver and fixed this small mutation, which then allowed the baby to make the enzyme that reduced their dependence on external medications and therefore is going to be life altering for that baby and for babies like this around the world. Now, why this is such a landmark is because this is the first time that we are seeing something made very specifically for a rare disease patient, right? So when we say rare diseases, it brings to mind a lot of different types of conditions, right? Neurological, blood diseases, muscular dystrophies, blindness diseases, hearing diseases, so many things. And they are usually neglected, right? Because from a large industry point of view, you want to first treat those who are thousands or millions in number rather than the rare disease. But if you put all the rare diseases together, there are millions of people who are either going to die or have very poor quality of life. So this brings in hope for all of those millions who are affected by this. That's why this is such a landmark.
Yeah. Thank you so much for that information, Dr. Debojyoti. Here, I would like to tell our viewers that for a very similar case, these two scientists, along with some others, of course, were leading a project and were very close to providing a similar treatment to a girl who unfortunately passed away while this work was going on. Dr. Debojyoti, could you like to talk about this particular case that you were working on?
Right. So, I mean, just around just towards the end of the COVID pandemic, we had this couple who visited my lab in Delhi and told that their daughter was suffering from as we call them, as a rarest of rare neuropathy, where a particular protein, because it was non-functional, was making aggregates in the neurons for a long period of time since her early teens, and it had now reached a stage where these neurons have stopped functioning, they are dead. And pathologically it was in a very, very critical condition. The patient was in a very critical condition, where essentially the patient was completely bedridden and not able to respond to any cues and so on. And this couple happened to be, it for them it was possible to actually be aware of the latest technologies in the field. And based on their knowledge of it, they had already started the treatment abroad in the US particularly, where initially they were on something called antisense oligonucleotide therapy, which was trying to give regular shots of small RNA molecules or small nucleotide sequences that would go and block the faulty RNA. But then when that did not work very well a long time, they heard that there was a treatment on the horizon called CRISPR, which could potentially create changes in the diseased gene, which would be like a long term therapy. So based on this information, they had gone to some of the best doctors and scientists in New York, who started working on this a couple of years before they came to us. But with the caveat that even if they would keep on working on it, the chances of a therapy in the US was very, very far away, for two main reasons. One was because they did not clearly see a regulatory route to getting it approved, and two, the enormous cost of doing this in the US, to make the clinical grade materials to treat the kid. So with this knowledge, they wrote to me. They came, both the parents came to me and said that, would you please help us with this condition. um, and I figured out that this was not a job for a single scientist in gene editing, because it was actually to create a therapy from scratch where none existed. And thanks to my friend over here, whom we were already collaborating on some other projects. So Orco essentially specializes on taking these molecules inside the body through very, very specific delivery vehicles. So even if I would make a gene editor for Uditi, in this case this girl, it still had to be delivered in the right place to be very efficacious. So I told the parents that me and Shobik, who are working in, Shobik is my collaborator in IGIB. We work together on this project. That even if we would make this, we would need another team to help us with delivering this into the patient. And that's when we made a kind of a group with Orco's team, with mine and Shobik's team here in IGIB. And we also over time received a lot of friendly support from other groups, including some in, for example, in Italy, who within just a week or so decided to give her antibodies, which she had made for several years, working on this kind of a disease. Because this disease was completely new to us, and it was not studied well. It's a very rare disease. So that's how it all started, and we decided to put all the resources at both labs in Bangalore and Delhi students and everything concerned to just treat this single individual. That would mean that we would have to make a model of her condition in the test tube. In this case, we decided to make stem cells. We actually did a process called reprogramming, where we would take her cells from her saliva, because she was already in a condition where she was drooling, and we took some saliva samples. We converted that into her own stem cells through a technology called reprogramming, and that now allowed us to actually mimic her condition in the petri plate, basically. And then we tested our base editors, which is the same technology which has gone into the study which we just discussed about. But this base editor technology was slightly different from the one that has gone into the study. It was based on a proprietary CRISPR system which we had just gotten an American patent for, and we believe this has a very high specificity and safety profile. So we made that, we compared it to the existing one, saw that they are comparable. Then we went into the cells, we did the gene editing, we did sequencing, we figured out that sequencing is fine. I mean, we can actually correct the sequence back to normal. Then we made neurons from that, from those cells. And once we made neurons, we saw that very, very promisingly, those aggregations that we were seeing could actually be restored back to normal upon gene editing. I think that was a kind of a little bit of a eureka moment, that there is a possibility we could cure, or we could develop a therapy. We don't know if we would cure, but we would develop a therapy. That was the time when Orko took up this at his end, and I think he's going to explain what he did over there. But essentially, just to summarize from our end, it was like a mission mode kind of for us, that there was only one person,
All this
and
to
that
save one life who was which was very precious, but unfortunately, in this case, time didn't support.
absolutely.
Yes.
Absolutely. We knew that we were, fighting kind of I would not say a lost cause, but we had a little bit of a hope, knowing that, sometimes, there are miracles that can happen in terms of when a lot of people put their efforts there. But we did meet, Uditi at some point in August, 2 months before she passed away. And I think meeting her was also a big turning point, for at least for me, definitely, to understand who this what is the condition that she is suffering from. And it only
Before
made
this, you hadn't met her yet. You'd been working on her case,
we
but
were
you
meeting
hadn't.
with the parents. We of course saw the reports and everything. We had seen her pictures and all, but to have that personal interaction was quite a changing, life changing moment for me, at least. And I think I don't want to impinge on what Orko has to say, what he did once he got these editors. So why don't you to tell them?
Yeah, yeah, sure. Please, Dr. Ghosh, you also share your experience of working on this project.
Right. Uh, so essentially, when like Debo said, he spoke to the parents, and his editors were a potential solution. But how do we get the editors into Diti's brain? That was the question that was posed to me. Now, that's where there's a couple of unique things that we do that came to the fore. So, the editors, the CRISPR editors, the enzymes that will go and make these very efficient small changes that will fix the DNA, these are enzymes, these are proteins, which are very large, very big. So the delivery vehicle that we chose to deliver the DNA for these proteins, these CRISPR editors, was a vector called recombinant adeno-associated viral vectors, okay, AAV vectors. Now, these vectors are the gold standard for doing gene therapy across the world. In fact, the first human gene therapies that were approved, which was for an eye disease, was an AAV vector. Thereafter, for blood diseases, for muscle diseases, a variety of rare conditions, AAV vectors are the vectors of choice to do that. However, the one of the drawbacks of it is that the vector is very tiny. So I'll use an allegory to explain what I mean. So let's say you cook a large pot of, say, rajma, right? Now, and you have a small fridge. So if you want to put it in the fridge, you will put it into two smaller boxes, let's say, so that it
Right.
fits into the fridge. And
Mhm.
then when you have the guests coming over, you take it out again, you put both of them together in the large pot, heat it, and then serve it, right? Now imagine this is happening to CRISPR. So the CRISPR is very large, and the delivery vehicle that we have, the boxes are tiny. So we split the CRISPR enzyme sequence into two parts, put them into two boxes. Now we put both of these boxes into the neurons, into the brain cells. And those two parts now come together and make the full gene again and makes the protein. So this involves a lot of engineering. So this is hardcore sort of biotech, or you can say genetic engineering that we do. So that was the aspect that we were doing. And we also, before this, we had not really worked on any kind of brain diseases. So we had to teach ourselves how to do all of that. Then we also needed to figure out, so the AAV vector, that comes in many different flavors. Okay. Now, whether the green one or yellow one, so we call them serotypes, AAV 2, 6, 8, 9. So there are different serotypes. So we tested them in mouse models where we delivered them through many different routes. We surgically put it in the brain, we put it through the spinal cord, we put it through the bloodstream, and tested which one goes to the brain most efficiently, because we are going to get only one shot. So we have to use the most perfect, the best system that we can potentially. So therefore, we had to put the delivery capsid, we had to choose the best one. We had to use this dual vector system. So then we created the dual vector system, and both in my lab and Devo's lab, like he pointed out, we took Uditi's blood cells, we converted them into iPSCs and then to neurons and things. So we tested them on her cells, and then we put from her cells, we, after validating what is going on, whether or not the two vectors are coming together, expressing, editing, etc. So that's the part that Debo checked in his lab, and he also found that wonderful observation that it can actually reduce the aggregates. So we knew that that is a problem that is happening in Uditi's cortical neurons, in her brain cells, in the thinking center of her brain. And that was the hope, that now if you use this dual AAV CRISPR editing system, this would have been the first instance of a dual AAV system in the world. Even in this current paper, there is no dual AAV system. They have used a nanoparticle, so they could use a single cassette. So the dual cassette system will still be a unique thing when it comes out, which will be the first in the world from our groups.
Yeah. So very interesting case, and for the viewers who might be interested in understanding it in a bit simple manner, also we had done a report, and in the description of this video we would put a link, so those of you who are interested can go through the link and read a little about this case. But very interesting things that you both mentioned. But one basic question, which probably I should have asked a little earlier, is we have been hearing about gene therapy for some diseases like cancers and other rare diseases as well. What exactly is it? Are these two things different, gene therapy and gene editing? If yes, how? Dr. Devojyoti, would you like to answer that?
Yeah, so gene therapy is a much broader term where you are essentially dealing with changes in the genes. Traditionally, historically, gene therapy was used synonymously with a technology called gene augmentation. Augmentation means that you add an extra copy. Either you add an extra copy into the genome of an individual, or you express an extra copy through some other mechanism, like in case of you put that in a virus, and that virus expresses that. This works because in most cases, if you have a faulty gene, if you want to express that from an additional copy of that gene, then that faulty gene, even though if it produces a misfunctional protein, the other copy is able to take care of it. Gene editing precisely is about doing the change in the DNA of the individual himself or herself. So there, it's not more about adding an extra copy, but rather it is about making the correction in the genome. or very precisely, gene editing always entails doing something to the genome of an organism. Gene therapy, although it's a broader term, still is used for meaning when there is an additional copy of the gene. So
Okay.
when you talk about, let's say, cancer, where the prevalent technology is, let's say, the CAR T therapy, which is really now very popular among a lot, and many people know about it. There too, it is a type of gene therapy, because there you are actually adding some additional copies through a lentivirus, essentially, which tends to get into the genome and put that additional copy into the genome. Um, in the case of Uditi, what we were doing was, it's a very interesting thing, because we were actually delivering the machine through the AAV, but the idea was to do a gene editing in the end. So we would have delivered the machinery through the AAV, it would have gone into the right cells, but then the machinery would come out of that AAV, if I can say it in that way, and then would have made the correction in Uditi's own genome and converted the faulty mutation back to normal. So it's a classic case of a gene therapy, traditional gene therapy delivery agent being repurposed for gene editing. And like Orko said, that it was a bit of an ambitious, very high, far-fetched thing that we were trying to do, because I don't think so this has been reported, or neither is in the progress anywhere yet. Mainly because the brain is a very difficult organ, very sensitive organ to reach, compared to some organs like liver and the lungs and so on, where typically sequestration of molecules tend to happen. So that was a big challenge. And I mean, of course, we took up the challenge because the intention was always to treat the patient, but it gave us a lot of learning lessons, and
Yeah.
we hope that with whatever we have gotten from there, it sets up a blueprint for targeting some
What is
other
coming
diseases.
next? Yes.
Yeah.
So a lot of work in this space is already happening in India also, and that's very exciting. This is very sophisticated. At one point, this was sort of futuristic, but it's happening now, and it's really, really exciting and may have profound changes. But I also want to understand. So we have been hearing about these things like designer babies, for example, or eradication of diseases. Do you think in coming times using these technologies is possible to have sort of babies who probably are the way you want them to be, or to eradicate certain diseases? Dr. Arkuf, would you like to take this question first before Dr.
Yeah.
Chakravarti answers?
So the question that you asked is a profound bioethics question that we are grappling with all the time. So yeah, the promise of when we first sequenced our genomes was, if you look back at the original papers and everything, was that we will create a pathway to eradication of diseases using the knowledge of genetics and genetic engineering. But that has profound ethical implications of it. So designer babies is one such ethical implication. So as of now, the law of the land states that we cannot make uh, genetic changes in natural healthy born individuals without any due cause. So if you have any disease, rare or not, but which has a genetic background to it, then you are allowed to make these changes to cure or help the patient. But if you want babies with, fairer skin, blue eyes, those kinds of things, that is not allowed, and that should not be, because I think then we will lose the genetic heterogeneity, which actually gives us the individualists in and makes us the individuals that we are, right? So from
Absolutely.
a bioethics point of view, this is definitely a no-no. This should
No.
remain within the realm and controlled strictly just for ethical clinical purposes. The other arm of gene therapy, which is a reality, unfortunately, in certain rogue nations and rogue clinics, is augmentation for like sports and other things. So you can get gene therapies that will increase your stamina, increase your muscle density, muscle mass, help you run faster, longer, higher, etc.
Okay. So work
So
is already happening on that front to
certainly,
create this kind of people.
some of the therapies that we create can be used in healthy individuals to do that.
Okay. So, Dr. Chakraborty, would you like to share your perspective on this as well?
Yeah, I think it's a very important question, but I just have a slightly different opinion on this, in the sense that to me it appears that the science of genetic engineering is very rudimentary still. We are really at a very
We
basal
are taking
level.
baby steps probably as of now.
Yeah, the reason for that is we do not understand, I think, almost 99% of how our cells and bodies function. So to say that we can accurately create designer babies, I think it's really out of the question right now, because even a simple thing like a skin color has hundreds of genes which
Okay.
actually cause that. So in reality, we do not even know how our bodies really work. We are trying to understand that. That being said, the bigger problem then actually the genetics and making such path breaking tools is the bigger problem than that is actually the accessibility and affordability of such kind of therapies, and that needs to be talked about much more. I would give a kind of a similarity to the iPhone, right? The iPhone is a fantastic device in terms of development, but it's not something that everybody has access to. Okay, so
Absolutely.
we would have a condition, or we would have a situation where there could be gene therapies that can potentially save kids or save individuals, but only accessible to a certain few. Now that becomes already a question of have and have not. That is even more dangerous than making a designer baby, because you have fitter individuals in the population because their genes have been fixed. But there would be also individuals who, just because they cannot pay for it, will not have access to that. So
Yeah.
these are discussions which are more, how to say, at the level where intersection of politics, intersection of the medicine, who benefits from it, what is the intention to provide that cure, all these come together. But it's
Yeah, so
essential.
that's a very interesting answer to a question which was very complicated. But at the end of it, any medicine, any treatment that you offer has to be accessible to people. In India, we
Exactly.
take pride in saying that we provide cheaper medicines, we provide cheaper vaccine to the entire world, not just India. So, since you two have been working on CRISPR in India, you can share the work that your labs particularly are doing. How far have you achieved so far, and which are the conditions that you're working on to our viewers? Dr. Arko, would you like to talk, because you work on eye diseases largely, right?
Yeah, so, but not exclusively. We work largely on eye diseases and muscle diseases. And
Mhm.
for brain diseases, this was the
As well.
one case that we worked on. And I worked with Debo on creating in vivo gene therapy for sickle cell anemia. So, to your question on accessibility, so I will answer this in two parts. So,
Mm-hmm.
um, certainly, our goal and our entire aim of creating a gene therapy laboratory with the best capability, equal or better than most parts of the world, that effort has been towards having indigenous gene therapies that can be applicable to our patient population. The two main reasons for it are accessibility. And gene therapy drugs are by far the most expensive man-made materials in the world, right? So uh, nobody can afford millions of dollars here in India to pay for a gene therapy. So we have to create indigenous technologies, indigenous processes to make these medicines, these genetic medicines, so that we can provide it to our patients for an accessible fee. I mean, it is still ongoing, but we have taken baby steps where we can now safely say that the cost of an equivalent gene therapy product here in India made by us will be at least 20 to 50 times cheaper, 20 to 50 times cheaper than the international cost, without compromising on quality, safety, or efficacy, all of them. So the way we have done that is by developing the technologies from the grassroots onwards here in India. So the complexity of these technologies is what drives the price of it. So in the Western countries, to develop these sort of technologies over, say, a 10 to 20-year period, the cost of manpower, infrastructure, etc., all of that put together, plus the cost of medical care for the patients suffering from these things, they put all of that together and come up with these amounts, right? These are crazy amounts, millions of dollars for a therapy. So we are sort of blessed in a way that we could leapfrog a lot of those development parts. We already had the knowledge. So we were able to create these technologies in-house and make it better than what's out there. And so what we have done now is that we will actually reach trials with a drug which you can have better activity for a lower dose,
Okay.
which means I make the drug which is
Mm-hmm.
more potent. So I have to give lesser amount of it to get the therapeutic benefit. So therefore my cost has come down, my side effects of the drug have gone down, so my efficacy of the drug is higher, and I am making it for cheaper. Now we are making it for cheaper because, let's face it, we are all cheaper as manpower, right? So that's one big difference from the West. Although we use 100% of imported materials and instruments, but still the overall cost then goes down. So that's how we are improving accessibility, by putting the cost down. Now, the other arm of accessibility, of course, has to come from the clinical part, which is still in development, because our clinicians and our hospitals should be able to deliver the
Yeah,
therapy.
absolutely. That's a very important point as well. They
Yes, so
have to be
that is still
trained to
something that needs to
be
talk.
able to deliver these medicines.
Exactly.
Dr. Debojyoti, I would put this question slightly differently to you. Like I mentioned earlier, medicines are being made in India, generic versions, largely the Me Too drugs. But there is always India's medicine industry is also, sometimes lambasted for not being able to innovate. Now, innovation is something that India lacks, despite of providing the large amount of drugs and vaccines to the world. Do you see India's laboratories in the near future innovating when it comes to gene therapies and these new therapies when it comes to life sciences?
Yes, I actually do see that, but I don't see it to the extent that we need. As in, our needs are far more than the number of labs which can cater to the variety of disorders, the variety of diseases, and the amount of development that is essentially happening elsewhere. So, if we have to keep that in mind, then we need to ramp up our R&D sector, because you are right that most of our big pharma companies, generally they are a little bit reluctant to add a lot of value to their R&D chain. It is more of
Yeah.
a production scale up and
Sorry to
manufacturing.
interrupt, but we always hear about this lack of collaboration between academia and industry. So that remains a challenge throughout, right?
That remains a challenge. There are two reasons for it. One reason is that there has been historically a little bit of a lack of faith on Indian R&D sector by pharma companies. Again, these are historical reasons. One of them is also because we have not, since the last end of the last century, we have not been at par in terms of new discoveries, new inventions. We have made significant ones, but let's say when we compare it to our US counterparts, or even across in China and so on, there has been a little bit of a lag, and that reason for that is the R&D for biology or for life sciences is not the same amount that is there required for physics, chemistry, and other sciences. R&D
Okay.
biology cost is way higher. It is a well-known thing, and the way that it was put forward in research grants or federally funded projects and so on. It would not, in most cases, allow this kind of a very high blue sky research, which was one of the reasons. And the second was the ownership of the ideas and innovations, which is very important for industries. Industries will only want to come in when they know that they have a safe, what they called as freedom to operate, fearing that there would be litigations, there would be, let's say, issues with legal entities later on, if they work with the Indian counterparts, where the technology belongs to somewhere else. So in this particular case for gene editing, gene therapy, I think both of us, particularly at IGI2, we have been focusing in on getting in as much FTO as possible. Not just because it's an important thing to do through patents and granted patents and so on, but also because it gives leverage to Indian industries to come and adopt them and have the faith that they can make their products themselves with this at the background. And I'm very happy to say that is happening actively, at least in IGIB. The IITs and ICES already have a good system for tech transfer, which is already existing. Not so much in the private universe, so much in the universities system, because there such kind of setups are often not there, and also in some of the smaller institutes. But it has to be promoted. It definitely has to be promoted.
Right.
Originality in idea converted into originality in an invention, which an industry can take up and then can innovate.
With that, we come to the end of this conversation. Let's hope that science progresses with all the ethical considerations in mind, and India also contributes to that. Thank you so much, Dr. Debojyoti and Dr. Arko. Really appreciate that you both could take time out and join us today. Thank you so much.
Thank you so much, Sumi. Thanks for having us.